Good evening, sir.
Good evening, my dears. Good evening, Ajay. How are you? How is going on?
Fine, sir. Everything is fine.
Okay. Happy Krishna Jayanti. All of you are
Same to you,
friends.
sir. Same to you.
Thank you.
Same to you,
Family
sir.
members. Thank you, doc. Thank you.
Good evening, sir. You can
Good
start
evening,
now, sir.
ma'am. Okay, ma'am. Thank you. Ma'am.
Yes, sir.
Sharing option, please, ma'am.
Okay.
Yeah. Thank
Yes,
you.
sir.
Yes, ma'am. Thank you. Thank you so much. Excuse me. Can you hear my voice, my dear students?
Yes, sir.
Yes, sir.
But here is you are not audible, sir. Like that. It
Sir,
comes.
till now it was not audible, sir.
So now you are hearing,
Yes, sir.
Okay. Last class, what we had discussed already, a little about. Okay. What is by vowels? What are the types of vowels? How they are classified according to their structures? What are the aspects of the vowels classified? Already you know a little bit, isn't it? They are all based on the position of the microfile with respect to phonicals and also salsa. These are the three pods. Under these three pods, they are classified into 6 types. Already we discussed, isn't it? See, orthotropous, anatropous, hemitropous, and campylotropous, and amphitropous, and the last one is called as cisnotropous. These are the things. Okay. See the diagram. These are the structures. Already we saw it, isn't it? Last class. What are the types of vowels? They may give match the followings, and sometimes they may ask the question, any one of the vowel structure they may give, and which one, this one X or something else, this is Y, this is also Z, like that they may, what is that, given as a specific, what is that, letters, and then you have to find out the letters in corresponding manner. It is also one of your, what is that, question paper. It may be direct diagrammatical questions, comes under the heading diagrammatical questions. Okay. See, so this is also same. So these are the structures. What are the examples has given? That is very, very important. So already we discussed yesterday these things, isn't it? Anyhow, once again we repeat these things and then enter into the remaining part of the lesson. Type of vowels with examples. See, autotrophs, atrophs, orthotrophs is an example, polygonum and piper and also cycus. These are the example. Anatropous, malvaceae, compositae, cucurbitaceae and solanaceae and castor. Okay. And campylotropous, cruciferae, leguminosae and also caparidaceae. These are the 3 things. Hemianatropous. See that anatropous is a structure already in our. Hemi means it's a partial, just like it's just a small bend like structure is there. Renanculaceae and primulaceae. Renanculaceae already we discussed. These things also. And amphitropous. Okay. Alimmaceae and butomaceae and centrospermiae and lemnae and also poppy. These are the 5th one. And the last one is called as cisnotropous. It is nothing but the cactaceae, otherwise called as opuntia. These are the types of vowels. These are the examples. Just to go through it, this one is a very, very important. They have given directly as an examples only. Okay. Some complication is there. More than one time you read it, you will get the answer clearly. Okay. Compare with the diagram also. Then only it will be better for your understanding. Okay. This is already we discussed. Okay. And on the basis of the integument, there are 4 types of vowels. Already we discussed. Integuments means inner part of the, just like a layer. Okay. So it is also modified in the form of fruit wall. The outermost layer, outer fruit wall, and then inner layer is called inner fruit wall. Okay. See that sometimes they may be, according to the number of the teguments, they may be divided into, see here, how many types is that? 4 types. Uni, that is consists of single integuments. Single integumented vowel. Bi means already, 2 integumented vowels. And tri means 3. And A technique means completely absence of integuments. This is also another specialized compared to the previous 3. This is a specialized structure. Particularly, you must know that what is that? Which one is the highest one? Highest number of technique. Lichi was an example. Okay. Sometimes they may ask the question, which one is the highest one? And also, which one is completely absent? Nil. just like the questions were said by the, what is that? Question expert. Okay. Then see the A technique in absence. Loranthus and Santalum. Okay. These are the two examples as given for what? A technique, absence of determinants. See. And specials also already discussed. Which one, the Carnicle and Strofoil. The Iferbaceae family, maximum, these are all are considered this. What is one? Which one? In a heavier bracillins, rubber family. Okay. Okay. And Arils, it's also Lychee, Maestrica, and Passiflora. These are the example. Perisperm already we discussed. Some unused nucellus present on the some seed. Okay. This is called as perisperm. And mostly found in black pepper and beetroot and Kenna and Mirabilis and Pinus Mirabilis. These are the examples for extra. That is called as perisperm. This is also sometimes they ask the real perisperm. Mostly they ask which family? Passiflora. Just note it down. Okay. See next. We are going to discuss about the after the what is that? Ovule development. What are the megasporogenesis? What is by megasporogenesis? See, the process of the formation of megaspore from the megaspore mother cell, okay, is called as megasporogenesis. Megaspore produced from the megaspore mother cell. That's why they have given us a MMC. Note it down. This point is very, very important. MMC. Mostly they ask the explanation. That means a expansion type of questions. You have to, for example, what is that? LED. Okay, what is by LED? Likewise, they may ask the MMC is nothing but the megaspore mother cell. Okay, come to the point. The process of formation of megaspore from the megaspore mother cell is called as megasporogenesis. What happened in this time during the development of ovule? Beginning of the new cells developed from the placenta in the form of a small rounded outgrowth like structure. That is the first initial stage. The megasporogenesis. Which one the ovule, how it will be developed? The beginning of the new cells developed from the placenta is the first stage. At this stage, what happened? At this stage, all the cells of the new cells or undifferentiated ovule or integument or like that. Homogeneous means what is that? All are same type of cells. Okay, and meristematic, and finally they become parenchymatous. First it is a meristematic tissues. Final what happened? It is also modified or converted or differentiated into parenchymatous cells. This means mass of cells surrounded by a single cell thick layer is called as epidermis. Listen here, the mass of cell is there. The mass of cell is also surrounded by a single celled outer layer is called as epidermis. So, total how many things is there? Epidermis covered within the cell, which one is known as parenchyma. Parenchyma is surrounded by a epidermis, like that. You might note it out. Okay. Next one. See, any one of the hypodermal cell of the knee cells is differentiated into increase in size. The hypodermal cell, what is that meaning of hypodermal? Before the epidermal cells, any one of the cell it containing cells is differentiated into increase in their size. Okay, it becomes different from the rest of the cells. Already we discussed it. Due to the presence of distinct nucleus, it is called as archisporial cells. Epidermis followed by hypodermis. The hypodermal cell, which one it has a nucleus, it differentiated into what is that? And grow as a new cells, but it is also different from the rest of the cells. That cell is called as archisporial cells. Okay, the epidermis is not a archisporial cell. Once again, archisporial cell is nothing but a hypodermis cell, but epidermis is there, not archisporial cells. That is very, very important. Okay, the archisporium divided. So this cell again divided mitotically to form an outer primary parietal cell and primary sporogenous cells. Again, the archisporials also further division takes place as a result, which mitotic means, body cells. This is also one of the body cells, isn't it? Okay. As a result, what happened? Parietal cell formation takes place, and also primary sporogenous cells. Okay. Then primary porogenous cells directly act as a megaspore mother cell. You got it, that points? The megaspore mother cell, how it will form, from the, which one, the archisporial cell. They may ask the questions. The megaspore mother cell is directly formed from which cells? Primary sporogenous cells. The primary porogenous cell maximum, it is also one of your archisporial cells. Okay, you have to write like as a equation: hypodermal, before that epidermal, then hypodermal. Okay, differentiated into archisporial, archisporial into primary sporogenous, primary porogenous into megaspore mother cells. Okay, this is the way you have to write as an equation. Okay, then what happened? It divides meiotically. After the megaspore mother cell formation takes place, it undergoes the meiotically to form a 4 haploid megaspore. That is very, very important. From the 4 megaspore mother cells, what happens? See that 4 haploid megaspore generally arranged in a linear tetrad. Already we discussed it, that things, isn't it? Suppose one cell divided into how many times? 4 haploids. The 4 haploid arranged in a linear manner from the top to bottom. Okay, so that condition is called a tetrad stage, similar to that of the, which you know, anther, male gametophyte. Male gametophyte is also a tetrad nature, but united with the callous tissues. When it undergoes a maturation, the callous tissue is completely degenerated. Likewise, but here what happened, the 4 haploid cells are produced. That condition is called as tetrad. Both the characters, both the male as well as female, there is a tetrad nature is there. You got it? Okay. See, these are the cells. Imagine, so this is the first cell. Okay, this cell is called as, what is that? What is that? Megaspore mother cell. It undergoes the meiotic cell division twice. You will get it 4 cells, just like a linear manner. This is megaspore tetrad stage. This is the, which one is the top? It is also micropylar, and micropylar is nothing but the ovule. There is a pore-like structure. See the diagram, then only you can easily understand it. Which one is called as micropolar end? See, so this is called as, okay, before that, see, yeah, so this one. Generally, the lower most of gelacell megaspore. This one is called as micropyral. No, when you are going to study the ovule structure, the uppermost, there is a pore, like there is a gap is there. That gap is called as, what is that? Micropyle, isn't it? Opposite of the micropyle, there is a gelasa. Okay, so here what they are telling is generally the lower most or gelacell megaspore. This one is called as lower. Okay, remaining functional out of 4 megaspore. This one is functional one. Remaining 3 is not a functional one. So they may ask the question: how many megaspore? It's not functional. Means 3 is not functioning, but the lower part only the functional one. Which one is the functional? Lower one. It is near to the, which one, gelasa part. These 3 are towards the micropyle. Okay, see that the 4 megaspore and the other 3 which lie towards the micropyle degenerated. These 3 micropyles towards the, what is the nucleus cells or completely divided up. Which one is remaining one? No, the cell part only the remaining one. Okay, this is very, very important. Okay, see that. So these are the structures. So gelacell only, what is that? Here there is only 1 megaspore mother cell. When it undergoes the meiotic divisions, what happens? It's divided into 2. And finally, again it will twice. Okay, you will get it the 4. What is that? Megaspore cells. So this is called as micropolar end. This area is called as, what is that? Chalasa end. Okay. See. Next, the function of megaspores produced female gametophyte. The functional megaspore, which one is at the last and least one is the near to the callosal part, you know, that functional megaspore produce female gametophyte. You note it down. This is also a important one. Megaspore produced from the female gametophyte. The most angiosperm, capsula, callosal megaspore remain function. Okay, all the angiosperms, the what is that? Megaspore always functional in nature. Remaining three completely absent in nature. Okay, so this is the very, very important thing. See that megasporogenesis. This is the first one. This is called as archisporial cells. When it undergo the next one, see the sporogenous cells produced. The sporogenous cell further again divided into so many. What is that? Haploid megaspore is the, see that 1, 2, 3, and 4 is the. From these completely 3 are degenerated. These 3 are degenerated. The only one is changed into the, which one is the megaspore. The megaspore already in our board divided into 2 nucleus. That after the formation of 2 nucleus, they move towards the pole. See that? And also they move towards the pole and also undergo the mitotic cell division twice. Okay. Here, one cell is the divided into 2. They can migrate towards the pole. This is a one pole. This is also another pole. They can migrate towards the pole. What happened? You know, again they may be divided into twice. You will get 4 cells. Okay, this is called as a, what is a mega gametophyte stage. They may ask a question: mega gametophyte, how many cells are present? 8. After the formation of 8 cells. Okay, here there is a one cell is there. Here also one cell. They migrate towards the center. Okay, central cells. They form together in the form of central stem cells. Okay, otherwise called as endosperm mother cells. So total how many is there? No, the one pole contains 3. Another pole is also a 3. But center there is a 2 cell is present. Otherwise called as, what is that? Central cell. Otherwise called as polar cell is there. You know very well about the embryo sac. This structure is called as embryo sac. Okay, the embryo sac. What are the changes which occur inside? The megaspore, only one nucleus is there and one cell is there. It divides mitotically to gives 2. Again, it undergoes the divisions and also move to the poles. Okay, after the reaches the poles, what happens now? To undergo the further divisions, it gives 8 cells. Again, here also 2 division takes place. Here also 2 division takes place. Totally 8 cells is there. From the 8 cell, from the 4 cells, one nucleus move to the center. Here, one cell move to the center. So 3, again 2 and 3. This is the arrangement of the embryo sac. Already you know very well about that things, isn't it? See. Then after that, what will happen? That things already discussed. You see that megagametogenesis. Megaspore is the first cell of female gametophyte already in our board. This megaspore grows in size and obtain nutrition from the nucellus. That things also how it will be get the megaspore, how it will be grow. Okay, they may ask the question how they get the food, how they will get the nutrition substances from the what is that gametophyte or embryo sac. Okay, see the nutrition from the nucellus. Each nucleus move towards the opposite pole and reaches at their respective poles. When it reaches the opposite poles, what happens? Both the nucleus lies at the poles, divided twice mitotically, resulting in 4 nuclei formed at the each pole. Total 8 nuclei. Out of the 4, 1 and 1 nucleus migrate from the both the poles. Okay, 1 nucleus from the callosal side and 1 nucleus from the micropolar side towards the center. They are known as polar nuclei. Note it down. This is very, very important. Okay, the both polar nuclei are present in the center. 2 nucleus present in the center. They are called as, what is that? Polar nuclei. See the structures exactly completed. Yeah.
Sir, could you explain that again, sir? Megagametogenesis.
Once again,
Yes, sir.
if you want, yeah, I will explain it. See that. See the megagametogenesis. Megagametogenesis is nothing but what is that? First cell of female gametophyte. You got it?
Yes, sir.
Okay. See the megagametogenesis. The megaspore is the first cell of female gametophyte, right? It gets the nutrition from the nucellus. Okay, after getting the nutritions, what happened? It can get the energy. It can move towards the opposite pole and reaches the respective poles. What happened? It undergoes the cell division. The division is called as mitotically. Then only you will get the, what is the 1 into 2, 2 into 4. So total 8 nuclei is possible. After the formation of 8 nucleus, the one from the which area? Zalazal side and one of the microbial sides move towards the center. So the center, there is a 2 nuclei present in it. The nucleus is called as, what is that? Polar nuclei. See the diagram, you can easily understand. See that here the only one cell is there, one nucleus, megaspore is there. The megaspore divided into 2. Okay, 2 nucleus here. One move to the zalazal part, one move to the micropyle part. Here given is this is called as micropyle end. So here the lower is the micropyle end. What about this end? Zalazal end, opposite side. Okay, here the zalazal end, this is the micropyle end. Okay, it's move on to the poles. Okay, after that the single cell divide mitotically twice. Here also 1 into 2, here also 1. Again it's divide means you will get 4 nucleus. Okay, this is 4, this is also a 4. From this 4, okay, move on to the center. Here the 3 only present in here, the 1 is move on to the center. Here also 1, move on to the center. See the center, there is a 2 nuclei. It comes from the one from the Salasa end, and this one comes from the this area end. Okay, so remaining things is called as, what is that? This is a complete structure of the pollens. Megaspore embryo sac. Okay, you got it that points. See here. So this center one is called as, what is that? This from this there is a center one is called as, what is that? Egg nucleus, center one. The both is present in the lateral side, Okay, that is called as synergy. Given here is a inverted manner. See here. Here the antipodal cell is here, here. So what about here, the antipodal cell is opposite. It may be inverted manner, they have given as a structure. No need to confuse, just to see the structure this only. After this, just come here. Okay, see here what happened, These are the, what is that? Salazal end. These are the microphylar end. Here there is a 3 nucleus is there. See that one? 3 nucleus is there. This is called as egg nucleus, and remaining 2 are the synergetic. Here, the center there is a, what is that? Polar nucleus, otherwise called as central cell. Okay, and then opposite means antipodal. Antipodal cells is present in the Zalaza end. Note it down, very, very important. Antipodal always present in the Zalaza end. And what about that synergetic as well as the egg nucleus present in the micropolar end? Because why it is present in the micropolar end, when the male gamete enter through the way of pollen tubes, mostly the micropolar end. Not only here, okay. Sometimes it may be Salaza, okay. Salaza end in this area, okay. And also finally it reaches here only and enter into the egg cells. So from here, what is that? There is a small protrude-like structure, isn't it? This is called as piliform apparatus. This is also very, very important. The piliform apparatus used for the, what is that? And allow the pollen tube through this way and reach the egg cells. This is the way. Okay, I will show you next diagram. You will understand. Okay, see here. These are the things. Remaining 3 nucleus at the each pole surrounded. Ajay, are you clear now?
Yes, sir.
Okay. by cytoplasm to form cell as result of cytokinesis. 3 cells are formed towards the micropyle, in which one cell is large and more distinct out of 3 cells. This cell is called as egg cell. Egg cell is compared to the other cells. Synergetic cell is there, no? The synergetic cell is very small one compared to the egg cell. Which one is the larger one is considered as a egg cell. And 2 smaller cells are also known as synergetic cells. Already we discussed it. Already we showed the diagram, isn't it? Okay. These 3 micropylar cells collectively known as egg apparatus. Once again, repeat for you. Egg apparatus consists of how many cells? See here, one egg cell. Sorry, here the spelling mistake. And 2 synergetic cells also there. These 3 is considered as a egg apparatus. Opposite side also have a 3 cells, isn't it? The 3 cells are formed towards and cells are called as antipodal cells. Both the polar nuclei present in the center is called as, what is that? Polar nuclei or central cell. This is the structure of, what is that? Embryo, mature embryo cell. They may ask the question, mature embryo cell contain how many cells, how many nucleus? Will you please, anybody else? Ajay or Dhanalakshmi or Ritika or whatever it may be, anybody else? The mature embryo cell contain how many nucleus?
Sir, 8 cells and 7 nucleus.
Very good. Yeah, correct. So mostly present in the which plant?
So angiosperm, sir.
Yeah, good. Angiosperm. Example? Capsula, isn't it?
So capsule. Yes.
So don't forget. Yeah, capsula. Otherwise called as typical angiosperm. Right. Okay. Just here. See here. This is the integument. Listen here. This is the integument. The outermost integument, this one, and innermost integument is also here. See here in the upper part, there is a gap. Is there no? Here the absence of integument. This area is called as micropyle. Got it? No? Okay. The center, there is a big, otherwise a large cell is there. The large cell is called as egg cells, and opposite side, the lateral side, there are 2 synergetic cells there. The center, there is a polar nuclei, otherwise called as central cell is there. These 3 are antipodal cell. This is the complete what is that? Embryo cell structures. This is also very micropyle cells. Okay, these are the 2 things. See. So this is the male gamete. This is one is the female one. Complete structure of the, what is the pollen grain as well as the megasporangium. Okay. Here how many male gametes are present? Total 2 male gametes are present. Okay. But here how many is the 8 nucleus? Okay. How many cells is there? 7. How many nucleus is there? 8. 7 cell, 8 nucleus present in the ovule, and 2 cell only present in the, what is the pollen grains. When it reaches the, which one, egg cells from the male gamete, what is my syngamy? Any one of the male gamete united with the female egg cells. That condition or that fusion is called as, what is that? Fertilization. What about the remaining one? It will enter into the another one that may be synergetic or somewhere else. As a result, the formation of endosperm. See, just see that this is also diagrammatical representations. Okay, so many times they were asked the question. See, this is called as micropyle, isn't it? So already we discussed it. This area is called as placenta. The placenta connected with the ovule, there is a stalk-like structure is called as funicle. This is the outer integument. This one is called as interintegument. This is, see, the outer integument. This one is called as interintegument. See, the egg cell is the large one, center. The opposite side, the lateral side, there is a synergetic cells. This outer covering of this 8 cell, there is a sac-like structure. That sac is called as embryo sac. Okay. So this is also a secondary nucleus. This one is called as antipodal. This one is, what is that? Chalasa. This is the entire structure of the ovule. Okay, micropyle, funicle, chalasa. These are the, what is that? Main basic concept for the types of ovule. Okay, anatropous, orthotropous, compylotropous, cystinotrophus, atrophus, like that. Okay, got it, that points. See, just before the fertilization, the unit are fused together in the center to form a secondary nucleus. definite nucleus. It is diploid in nature and one in number. The center there is a what happened? The both are united, a single cell. Okay, that is called as the two nucleus united and form a single cell. That's why it is called as what is that? One number. Okay, but the condition is 2n state because two nucleus haploid nucleus united. Okay, see that after mitosis in megaspore 7 cell and 8 nucleated structure is formed. This is very, very important. Mature megaspore contain 7 celled and 8 nucleated structure is formed. The 7 celled and 8 nucleated structure is called female gametophyte or embryo sac of the angiosperm. This is the very important mature female gametophyte. It contains 8 nucleated structures and 7 celled. They may ask the questions. So it is otherwise called as what is that? Polygonum types. Why it is called as polygonum type? These type of what is that? The 7 cell and 8 nucleated structure of the embryo sac mostly present or found in where the polygonum plant. That's why they have given us a polygonum and what is that type? This type of embryo sac is known as polygonum type because it is discovered by Strasburger in polygonum plant. That's why they have given us a name is called as polygonum type. Which one is polygonum type? They may ask the question like this: a female gametophyte or the embryo sac. Like that, they will not ask. They will ask the questions. The other word, polygonum type of embryo sac contain how many cells, how many nucleus. They may ask the question like that. So you have to remind this at the time of examination point of polygonum. It's nothing but one of the angiosperm. Okay, it contains this thing. So normally all the angiosperm they are having these type of what is that female gametophyte. But first it was discovered by the Strasburger in the polygonum plant. That's why they have given as a polygonum type. Okay, got it? Go to the next. And polygonal type of embryo sac is most common type in angiosperm. That is called as capsula. Polygonal type is also a common one, but it's mostly present in the angiosperm. Already told you, mostly in capsula. Okay. Then polygonal type of embryo sac develops from a single megaspore. Already we discussed it. The 8 cell, how it comes? It is also originated, developed from the mitotically from the single cell. So it is called as monosporic embryo sac. That is very, very important. The polygonal type, already these types also develop from the single megaspore. This is also sometimes they ask the question. So it is called as a monosporic embryo sac. Okay, then already discussed. Finger-like structures are produced from the outer wall of the synergetic or known as piliform apparatus. I told the piliform apparatus allow the pollen tube through the way of what is the microfile and reach the what is the male gamete into female gamete, otherwise called as egg. With the help of these structures, synergetic absorb food from the nucellus and transfer to the embryo cell. Not only the filiform cell, they will not allow not only this function only, and also it is allow the what is the absorb the food material from the nucellus and transfer to the embryo sac. A filiform cell apparatus also used for the which one transport of nutrition for embryo sac. Okay, piliform also secreted chemical which attracts and guide the pollen tubes. This is also another special type of character. These are the three things: is that which type of which structure, which shape of the structure or finger-like structure they told that is called as piliform. It is originated from where synergetic outer wall. You note it down. That is very, very important. And then it's produced absorb the food material from the nucellus and transfer to the embryo sac. Third one, it secretes the chemical substances. It attract and also guide the pollen tubes. These are the very, very important things. See this is the structure already we saw it, isn't it? These are the main things already. These are the structures. Okay. Next, the interesting topic is that is called as pollination. Okay, so last up to this level, what are the things we studied? First, what are the parts of the flowers, and Anther, how it will be produced? Microsporogenium, genesis, microspore, structure of anther, inner side. What are the changes which occurs? All those things we will study. Then enter the megaspore. Megaspore is the female gametophyte. How the megasporogenesis started? How the single cell is converted to the megaspore mother cell? How it will be developed? Finally, it reaches the complete mature megaspore, otherwise called as mature. What is that? Gametophyte. So both are ready. This stage is called as, what is that? Juvenile stage. When you are studying the first term, before the maturation, before the pollination process, what is that? That stage is called as, what is that? Juvenile phase. After the maturation, they are ready to, what is that? Pollinations. Undergoes the reproductive process. It is also called as a third stage of the plant life cycles. So see here, pollination, I didn't know very but transfer of pollen grains from the anther to the stigma of a pistil is termed as a pollination. This is already know very but otherwise called as, what is that? No, both male and female gametes are united or non-motile nature. Here the pollination I have given, I have chosen a slide. See that these are the two plant flowers. Sorry, the same flowers but different branches. You can imagine like that, or different plant or same flower. Okay, same branches. See here the pollen sac is there from the anther. Pollen grains are there. The pollen grain transfer through the way of vector. What is that? This is also honeybee transfer to the another plant. This is called as cross pollination. We will see one by one. See the next slide. See. So what is my pollination? I already discussed it. Any one of them, pollen grain transferred to the stigma. Okay, otherwise called as female reproductive organ is called as pollination. Okay. What are the kinds of pollination? Pollination already discussed. There are two types of pollination is there. One is called as self-pollination. Another one is called as cross-pollination. Self-pollination, what is the meaning of self-pollination? Suppose the flower is there, here the male gamete, here is the female gamete is there. See the diagrams. Here the male, this one is called as male gamete, this one is called as female, isn't it? Here, the single flower contains both the, what is that? Reproductive pod. Here what happened, autogamy or self-pollination means the anther produce the pollen, it is reaches the same flower of the, which one, female pod is called as autogamy. See here, the next, see here, this one, structure. This is the same flower, isn't it? Same flower is that, okay. Say this is the female. This one is called as male. Okay, this is anther. When it undergo the dehiscence process, it releases the so many pollen. Number of so many, what is that? Pollen grains are released. It reaches the same flower stigma. That condition is called as, what is that? Autopollination or autogamy, otherwise called as self-pollination. Okay, self-pollination, simply called as self-pollination. The self-pollination further divided into autogamy and geitonogamy. Once again, autogamy and geitonogamy. The autogamy further divided into whether direct or indirect. According to the direct or indirect, so they divide into direct autogamy and indirect autogamy. Once again, repeat for you. Pollination divided into two, self and cross. The self-pollination divided into autogamy and geitonogamy. The autogamy depends upon the, what is that? Involvement, direct or indirect, like that, divided. Okay. Here, next one, the cross-pollination. Based on the plant, they may be divided into two. That is called as allogamy or xenogamy, otherwise one called as hybridism. And another type of pollination is also possible. That is called as hybridism. Cross-pollination divided into based on the plant, based on the factors. Factors means what is the, they may be involved in the, what is the abiotic factor and biotic factor. Okay. Cross-pollination based on the plant, based on the factor. Based on the plant means allogamy, otherwise called as xenogamy, and hybridism. Based on the factors means there are two factors is there. What is that? Abiotic factor and biotic factor. Here, see here, these are the, what is that? Hydrophil and animophils are considered getting known as biotic factor, or abiotic factor. These are all are abiotic factor. Zoophilic is a biological or living organisms that is called as zoophilic. Okay, this is the kinds of pollinations in your book. Okay, see that. Autogamy, already told you. Pollination within the same flower, okay, is called as autogamy. It open and exposed anthers, stigma. Autogamy is rare, where the viola, common pansy, oxalis, and camomile produce the two types of flowers. These are another type. Pollination already the same flower. Okay. Suppose if it is exposed to the anther or stigma, what happen? Autogamy is rare. Okay. See here, according to these type, there are two types have been reported here. What are the cosmogamous? Exposed anther and the stigma. This is called as cosmogamous. Clistogamous, closed anther and stigma. This is also another. They may ask the question. Clistogamous mostly they ask. Okay. The clistogamous means the closed anther and stigma means what happens? Self-pollination will be possible. Exposed anther and stigma means what happens? It is a cross-pollination mostly possible. Okay. See, clistogamous flower is invariable, autogamous, and assured seed set even in the absence of pollinator. Clistogamous flowers, what happen? Invariable autogamous and assured seed set even in the absence of pollinator. Pollinator, without pollinator, can produce their seeds like that. See the diagram? So this is a self-pollination, isn't it? The same stigma is also a same flower, and pollen grains also, otherwise called as anther, is also same flower. The anther transfer from the same flower. Okay. Okay, stigma. When it reaches stigma, automatically it will be entered in the pollen tubes formation takes place. The adversarial needs from this area. Okay. And also the exine entine is also there. Okay. The intine produce a pollen tube like structure. When also it move on to the which one Zalasa side, and also the wall area, and also microphyll side. We'll see the one by one. So that things how it will be entered. And second type of pollination is called as geitonogamy. This is very, very, very important. Pollination between two flowers of the same plant. Okay. Pollination flower, the two flowers of the same plant or same branches or same plant. Okay. Pollinated by pollinating agent genetically similar to the autogamy. Most of the geitonogamy, what is that? Genetic is similar in nature. Example, cucurbits. Okay, cucurbita family. So this is very, very important. Geitonogamy, the pollination between the two flowers of the same plant. Okay. So pollination by pollinating agent. What is meant by agent? It's agent may be insects or wind or whatever it may be. Okay, pollinating agents. Genetic is similar to the autogamy because autogamy means similar genetical structure only produced. Okay, this is why cucurbitas. The last one is called as genogamy. Transfer of pollen grain from the anther to the stigma of different plant but the same species. Take an example, always a family is here. Okay. And then after 10 yards, there is another species also there, The pollen grain transfer from the nearest your place to the 10 feet again, the opposite species. They can transfer the pollen grain from to here to there. Okay. This is called as what is that, cross pollinations. It brings genetically different types of pollen grains to the stigma. So these are the two, what is that? Differences between the geitonogamy and genogamy. Genogamy means different type of pollen grains. Here, there is no different type, only the same type of pollen grains. Genetically different, but here genetically similar. That things you should know. Okay. So it is also very, very important. See that this is self-pollination. This is the same and the same flower. But cross-pollination, it may be in the same branches. Okay. Okay, same branch or different branches. That is called as cross-pollination. This is the one flower. Here the anther is there. It transferred to the another flower of the, what is that? Stigma, female reproductive part. So this is called as cross-pollination. Suppose if it is another species also, another species, it is also, what is that? Genogamy, like that. It comes because It is a one type of anthers. It is another type of species, Malvaceae family and Solanaceae family. Okay, it can cross with anyone, female to male or male to female, like that. Okay, so that time what will happen? No, a different species, different genetically different plant you will get it. Okay, see the entire diagram in the one entire three type of what is that? Pollination takes place in the single plant. See here, see autogamy, the same flower, the anther and what is that? And pistil as well as the anther and pollen grains transfer to the same flower stigma. That is called as autogamy. What is the meaning of geitonogamy? The same species but different flower. See here, this is called as geitonogamy. With the help of the what is that? Pollinator. Here the pollinator gets given as insects. Okay, it's nothing but I think small pedils or something is butterfly like that. Okay, they have given. Okay, and genogamy. See that genogamy, the one plant to another plant. Here one flower to another flower, but here one plant to another plant. This is also called as genogamy. You know the difference between these three? Can you hear the? Okay. Can you easily differentiate these three things? Any doubt in this area, ba? Students. What is my autogamy? What is my autogamy? It's nothing but the same flower only. The male gamete transfer to the female gametophyte. Only the single flower only. But geitonogamy, the same plant but different. What is the flowers? But genogamy, the one flower of the plant to the another flower of the another plant. Got it? No? See that. So they may ask the question: unisexuality of the flowers prevents the question. Okay, the geitonogamy but not genogamy, autogamy and the geitonogamy. But the answer is autogamy but not geitonogamy is the correct one. Unisexuality. Okay, both geitonogamy and genogamy. This is also so many times asked, autogamy but not geitonogamy. It's okay. Check it once again. So the next one. So what are the agents of pollination? Suppose the male gamete as well as the female gamete fuse together with the help of the mediator. Already we discussed. Okay, according to the factors, there are two have been reported. One is called as the abiotic factor. Another one is called as biotic factor. Already I told you. The abiotic factor is nothing but the non-living things. It is called as wind and water, as given as an examples. Okay, biotic factor means insects, bats, and rats, reptiles, and also mammals. These are the things as given to the biotic factors. We'll see one by one. See here. So these are the agents of pollinations. Can you see the diagram, pa? Chart?
Yes, sir.
Okay. See that agents of pollination here, the divided into two. One is called as the abiotic factor. Another one is called as biotic factors. The abiotic factors just see the wind, otherwise called as what is that? Wind, air. Okay. Here water or hydro. Okay. So biotic factor just see this bats, insects, and reptiles, and also mammals. These are all considered the biotic factors. These are the abiotic factor. See. Pollination. Already we discussed these things. According to the pollinations, according to the factor, they may be divided into many things. See that pollination, self-pollination, cross-pollination. Same flower and same plant, different flowers. Cross-pollination, genogamy. It is also the pollinating agent is biotic. Okay. Biotic means living organism. What other things is there? Snail. Okay. Snail, insects, birds. Okay, and bats, and also other vertebrates. Okay, these are all considered living organisms. The same snails, see that one by one I will show you. The abiotic factor is 2. The wind and anther to stigma by wind is called as anemophilic. Suppose if it is water is called as hydrophilics. Okay, see. See, so these are the things. Abiotic agent, anemophilic, hydrophilics. Biotic agent, endomophilic, ornithophilic, chirotrophilic, malacophilic, and myrmecophilic, zoophilic. They are all are considered zoo because it's all are different, are disposed by only the animals. That's why they have given as a zoophilic. Living organisms, even human beings also, they may spread. Okay, see that. So suppose the pollinate, yeah.
So what is this malcophily and myrmecophily?
Yeah, I will tell you. I will show you. I will show you. I will show you. Okay, the diagram will come after this slide. Okay, okay, I will show you. Adaptation of flower for the pollination. What are the adaptation? Then only the pollination will take place. For example, anemophilic flower is the how it will be. Okay, that wind pollination is nothing but what is that? Anemophilic in nature. The pollen grains light and non-sticky winged one. Then only it can be easily transferred by where? Wind or air. Anther well exposed. Stigma large and feathery nature. Flower one ovule arranges as the inflorescence. Example, corn and cob. Wind pollination is called as anemophilic. Wind means inhourable air. What are the flower characteristics? Then only the plant undergoes the wind pollination. Otherwise, it is not possible. The pollen grains light, non-sticky winged. The anther well exposed. Stigma large and feathery. Then only it can be easily fly on the air in the atmosphere. Okay. Flower one ovule arranges as the inflorescence. Example, corn and cob. See here. So this is called as a wind. Okay. See the feathers as well as what is happening. It is a lightweight is there now. It can be easily spread through the atmosphere. Suppose the air is passed on this way, So likewise, okay, the feather-like structures. These are the what is the male gametophytes? It transfer from the one to another one. This is also an example. See, water pollinations. Water pollination, otherwise called as hydrophilic in nature. The pollination by abiotic agent like water. This type of pollination is very rare. About 30 genera, mostly monocot. How many genera? 30 genera as given. Example, Wallisneria, Hydrilla, and Justera, and sea grasses. These are all are considered getting known as hydrophily because these are all are hydrophytes. They only lives in the water under pond or whatever it may be. Okay, pond or river or sea like that. Wallisneria. All the plants are hydrophilic nature, hydrophytes. That's why it is called as what is that? The pollination takes place through the water molecules, are called as hydrophily. Pollen grains are protected from the wetting by mucilaginous covering. This is also a special features of the water pollinating agents. Okay, just imagine, suppose the hydrophytes produce the pollen grains, easily wet on the which one, water. What happened? It will not reach as a stigma. It will not produce a what is that? Gametes. Okay, so that's why the mucilaginous covering is also protect the which one, pollen grains. So it will not easily what is that? Dissolve in water molecule. That is very, very important. Already you know about that things. See, so this is called as pollination of water by Wallisneria. This is a female flower. This one is called as a what is that? Male flower. This one is male flower. Okay, it is a what is that? Easily floating on the surface of the water. It can be easily transferred from the female. Okay, flowers. When it reaches the top, what happens? There is a wind. Okay, it will be reach the male flower to female flower. See that? Main features of the wind and water pollen. Doubt you have. Okay, see that. Produce pollen grains in large number. So main feature of wind and water pollinating plants, the pollen grains is large number. That means 100,000 millions like that. It's a microspore. Do not produce nectar. There is no nectar gland present in it. Completely absence of nectar. Example, Wallisneria. Because, okay, so it is a, what is that? Light white. Male flower or pollen grains released on the water surface carried by water some eventually reach the stigma. These things already you know, but already we discussed the diagram. Okay. Jostira, female flower remaining submerged. Submerged means it's a shrink into the water molecules in nature, but pollen grains long ribbon-like structures release inside the water, carried by water currents to reach the female flower. This is a special type of what is a pollination takes place under the water, submerged. Okay, they may ask a question. Okay, water pollination, which plant is submerged in nature? Justira. Ribbon-like structure, you can tell us the Justira. Okay, these are the things. See that? So this is called a female flower, male and female flower plant pollination area. Already we discussed these things. This is the top layer. Okay, this is the top layer. This is submerged plants. And female flower is here, and male flower also release the what is that pollen grains when it reaches the top, and also it automatically touches the it reaches the stigma of the female flower. Okay. Okay. See that. So now we are going to discuss one by one. First one is called as what is that? Hydrophilic, anemophilic. Next, endomophilic. This is also another one. The insect pollination is called as endomophilic, because endomophilic means you must note it down, very, very important. Flower should be how? It's a large in size, at the same time, it's colorful one also. Excuse me, ma'am. Hello, ma'am. Okay, ma'am, I will finish it now. Okay, okay. Okay, students. Okay, these things just left it here. Okay, the class is over now. Up to this level only, phytophytes as well as a anemophilic and also hydrophilic only. Just we leave this place. Okay, leave this class. After that, we will see it. Okay, if you have any doubt, please ask me. We summarize all those things. What are the female gametogenesis, and what are the how it will be ovule formation, what are the type of ovules? These things already we discussed, isn't it? Okay, thank you for the participation, and thank you so much for your time. Yes, thank you.
Thank you, sir.
Thank you. Thank you.
Thank you,
ma'am.
sir.
Thank you, ma'am.